This practice consisted in three activities. The first activity aim was based on acquiring the knowledge of which are the materials and equipment that form part of the genetic engineering laboratory, where are located, and their respective functions. The second and third activity aim was to analyze the accuracy and precision of the micropipette. From the results reported in tables, statistical data was obtained and then plotted. These results showed a low accuracy far from the actual value by up to 25% and high standard deviations up to 15, possibly due to the bad pipetting techniques. Knowing the material and laboratory equipment is a very important factor when you want to carry out a practice since it optimizes the procedure, avoids accidents, therefore it favors the care of the material and equipment. This is why the objective of this practice is focused on knowing and securing the genetic engineering laboratory material, starting with how to properly use a micropipette. Micropipettes can be classified according to the volume taken and by their mechanism and purpose. When working with certain fluids, the design of the micropipette employed may contribute to the correct sampling of the volume needed and its deposition. These mechanisms are air cushion pipettes, repeat micropipette, and positive displacement.
Activity 2. Transfers the following amounts of water, low, intermediate, and high volumes with micropipettes from 20, 200, 1000 respectively, from 2 to 1000 microliters. For each sample, the volume deposited in the aluminum paper of previously tried scale was reported. The high volume relationship was made with the water density, 1 gram per microliter. The volumes were recorded by repeating the measurement 10 times and taping the previous header each time. It is repeated 3 times for each micropipette with the 3 different ranges in the corresponding micropipette.
The second activity consisted in apply the good pipette practice and analyze the accuracy of the micropipettes. It was critical to adjust the volume correctly in order to minimize the error in the measurement. First of all, the attachment of the micropipette tip has to be appropriate according to the micropipette and the volume to be recovered. Then, it was taken a specific volume of water in triplicate and it was kept in a microtube of a corresponding volume. For a good measure of the volume, it's necessary to place the micropipette in 90 degrees of the water and avoid get too much into the liquid. These advices are useful to prevent more water than the calculated get into the micropipette tip. It is also important to know the general data about the fluid that it's going to work with, to use the best technique and minimize the error, for example, when a liquid is too viscous as glycerol and the air chamber of the micropipette cannot pull in easily the liquid. For better results, it's advisable to keep the micropipette tip around 3 seconds after the piston of the micropipette is completely loose into the liquid. This technique helps us to guarantee that the volume is being recovered proximate to the real volume. Then, the volume controller was adjusted to recover all the expected liquid that was kept in the microtube. The final volume obtained was recovered assuming the volume dispensed by the micropipette was accurate in each pipette. Knowing the expected value and the real value of the measures, it was possible to analyze the results using statistical tools.
Results. The results obtained from Activity 2 are reported in this part. Those are on the left side from team 3 and on the right side on team 4. Those are the different weights from the volumetric samples made from each micropipette, average and the standard deviations that were calculated using this formula. So the results, to make them more visible, we made them with graphs. So first we reported the weight on each measurement made on the tables respectively from each team, and then to interpret the results we made graphs. So the first graph in here shows volume obtained by each measurement using different micropipettes, and here are the volumes that we did in each part. And the second is the deviation, and in here we can observe the places where the deviation gets higher than in others, which is also reported in here with the green and red colors. And both tables and graphs we can see the same information, but obviously it gets different as it was made from different teams. On the results from activity 3, which was the recovered volume, the micropipettes that was made 3 and in the last 4 times. They show different volumes and their standard deviations also, where we can also see when it was lower and higher. And we have in here 2 graphs comparing both teams' volumes obtained, in which we can see where it was higher and or most representative. And the last graph shows the total volume recovered and their deviations, where it was higher in one part and lower in the others, depending on each team.
In practice, the material and equipment used in the genetic engineering laboratory was identified, as well as its proper use, application, and location. Specifically, we worked with micropipettes of different measurement ranges and carried out measurements of different volumes in order to learn how to properly use the micropipettes P20, P200, and P1000. Once activity 1 was concluded, statistical data were calculated as the mean and standard deviation, which is a measure of dispersion that indicates how scattered the data are with respect to the mean. Therefore, when looking at Table 1 and Table 2, it is possible to notice that the highest recorded deviations were presented in the 20 microliter measurements with the micropipette P20 and in the 1000 microliter measurement with the micropipette P1000, yielding values of 0.075 and 0.0099 respectively. Results that could have been influenced because of the bad pipetting techniques, which are usually one of the reasons for variations in volume. It can also be observed that the data in Table 2 are quite accurate and precise, since the values are very close to the real data as seen in the average, while the standard deviation shows the accuracy of the volumes obtained. In the case of Activity 2, the same statistical calculations were made, and a significant increase was observed in the values of the standard deviation with respect to those calculated in Activity 1. In this case, in Table 3 corresponding to the third team, a maximum standard deviation of 27.50 was obtained for the volume recovered with the micropipette P1000, while in Table 4 corresponding to the results of the fourth team, a maximum standard deviation of 15.38 was recorded for the volume measured with the micropipette of the same capacity. It's important to mention that for both devices, the maximum standard deviation values recorded were obtained when using the micropipettes for the first time and with the highest capacity, which indicates a need for improvement in the pipetting technique. Paying attention to the most common errors at the time of handling the micropipette, which consists of pipetting at an angle, choosing the tips incorrectly, submerging the tip too much into the solution, working too fast, etc. In addition, it should be considered that the maximum and minimum measurement ranges of the micropipette can provide a certain percentage of error. In comparison with Activity 2, the data of Activity 3 of both themes are quite inaccurate, since they are far from the real value up to 25%. And it can also be seen that they are not accurate, since the deviations are quite large in most of the data groups.
In addition to the bad pipetting techniques, another of the aspects that could alter the measurements of the volumes with the micropipettes is a poor or no calibration of these. According to the National Standards Institute, the calibration concept refers only to the action of comparing the reading of the measuring instrument with respect to the pattern with a known value or dimension. For this, a standard is used that ensures that what is being measured is correct. In this way, when verifying that the instruments were subjected to an adequate calibration process, we avoid that these become another source of error when carrying out the measurements.
On the other hand, the adjustment allows to improve the conditions of the measuring instrument and should not be confused with the maintenance or calibrations, since it can refer to a mechanical action, electronic, or by using a software to modify the measurements made and that precisely fits the pattern used. In the international metrology vocabulary, it is said that an adjustment of a measurement system is the set of operations performed on a measurement system to provide prescribed indications corresponding to given values of the magnitude to be measured.
The knowledge of the security measures, as well as the function and adequate use of the materials, equipment, and substances used in the genetic engineering laboratory will allow a correct manipulation of the instruments in future practices, which is useful to work efficiently inside a laboratory. In addition, the mastery of pipetting techniques and methods will allow to obtain accurate results and therefore a correct interpretation of them.